Welcome to the Mitosis Experience. Mitosis is part of the cell cycle. The cell cycle is made up of 4 stages. 3 of these stages, G1, S, and G2, constitute interphase. During the S phase, the human species' 23 chromosomes are replicated to create 46 pairs of sister chromatids. Mitosis, the final stage of the cell cycle, is the process by which a cell divides into 2 identical daughter cells. Mitosis itself consists of 5 stages. Before talking about the stages of mitosis, we should mention the mitotic spindle. The mitotic spindle is a bipolar array of microtubules that eukaryotic cells use to separate chromosomes. There are 3 types of microtubules present in mitotic spindles. The first are interpolar microtubules, which have positively charged ends that interact with the positively charged ends from the opposite spindle pole. The second type are astromicrotubules, which interact with the cell's cortex and position the spindle. A cell's cortex is a protein-rich layer of cytoplasm on the inside membrane of the cell. The third and final type are kinetochore microtubules, whose positively charged ends connect to sister chromatid pairs at the kinetochores. Kinetochores are large protein structures at the centromeres of each sister chromatid. Centromeres are where 2 sister chromatids connect. The first stage of mitosis is prophase. In prophase, the chromosomes, which are originally spread out throughout the nucleus, condense in the center. Additionally, the mitotic spindles assemble between 2 centrosomes or opposite poles of the cell. The centrosomes are structures that facilitate the mitotic spindle. The second stage of mitosis is prometaphase. In prometaphase, the nuclear envelope breaks down and sister chromatids attach to the microtubules at their kinetochores, which move the chromosomes to the center of the cell. The third stage of mitosis is metaphase. In metaphase, the chromosomes assemble in order along the equator of the mitotic spindle. Certain microtubules attach sister chromatids to opposite poles of the spindle. The fourth stage of mitosis is anaphase. In anaphase, the sister chromatids are separated synchronously. This forms 2 daughter chromosomes from each chromosome pair, which are then pulled towards opposite spindle poles. The fifth and final stage of mitosis is telophase. In telophase, daughter chromosomes arrive at the spindle poles and decondense. At this point, the nuclear envelope forms around each set of daughter chromosomes, creating 2 new nuclei. After telophase is complete, cytokinesis divides the cytoplasm into 2 separate daughter cells. Docetaxel, or Taxotere, is a strong chemotherapy drug used to treat many types of cancer. Chemotherapy drugs target cells that divide rapidly and kill them, and this makes them an effective treatment against cancer. Docetaxel causes mitotic catastrophe, which is where mitotic drivers activate where they aren't supposed to. This occurs because docetaxel causes deficiencies of mitotic spindle checkpoints in place to limit abnormal cell division. Without these checkpoints, chromatids may not line up correctly during metaphase, leading to cell division that creates cells with damaged DNA or chromosomes. Normally, these cells die. So in the case of cancer cells, docetaxel often kills off cancer cells, reducing the spread of cancer.